alaikum wa rahmatullahi wa barakatuh. I am Tehani البلوشي and I am a student studying abroad in the United States of America. I currently hold a Bachelor of Science in Chemical Engineering from the University of Long Beach in California. This video will serve its main purpose to support my application for the 2018 Omani's Ambassador Student Award. In here I will illustrate the contributions that I provided either to the university itself or any external academic platforms such as conferences. So without further ado, let me show you our lab and what I exactly did. This project was a part of a biomedical and biomaterial lab organized by Dr. David Stout, a well-known PhD researcher from Brown University. His lab is also supported by the National Science Foundation and the NIH Build Association here at CSU Long Beach. As far as my project goes, I had the privilege to work and investigate the impaired migratory behavior of neutrophil cells during sepsis conditions. Neutrophils are a type of immune cells in our bodies that travel to sites of infection during invasive attacks from microorganisms such as bacteria and viruses. It is known that patients with chronic diseases show that their neutrophils are non-responsive during these invasions and thus have a high mortality rate. There are many researchers that are dedicated to reveal the mysteries of why this happens. My project does exactly that. The project is sectionized into two different categories, both experimental and computational. The first phase involves creating a 3D biomimetic system that best emulates the extracellular surroundings of our cells in our body, which mainly proteins. And to do exactly that, we developed a collagen biomaterial matrix that hosted most of the experiment. The matrix was supported by a top and bottom housing for structural support. The second phase of the experiment was to introduce a dual chemoattractant signaling hierarchy system into the matrix. For those who are not aware, chemoattractants are chemical signals that induces chemotaxis in our bodies. It is a state of when cells respond to different signals present in a provided situation. The purpose here was to select two chemoattractants that best resemble signals simulated from invasive microorganisms like material properties, physical parameters, and molecular weight. We worked with rhodamine and interleukin as our chemoattractants. They both were allowed to diffuse into the matrix system to create a dual diffusion biosystem. Then, neutrophils were injected and their migratory responses were evaluated. The second category of the project was to simulate the process in a computational program and develop a MATLAB algorithm. The algorithm processed the image that were taken via a confocal microscope to study the patterns of displacement, concentration, and migratory forces. The project was also selected to be presented at the annual Build Symposium twice at CSU Long Beach and at the Conference of Women in Science at the University of Nebraska-Lincoln, where I had to meet with great scholars and share my work to the biomedical community. So all in all, to wrap up the video, I would love to give a special thanks to everyone that is behind the scenes for this award, including staff or sponsors. Also, I would love to give a special gratitude to the Ministry of Higher Education, because without their constant support to all of us, we would not have been here pursuing our dreams. So thank you so much, and I really hope that you enjoyed this video as well as you've learned some new facts here and there. For those who are actually rating my work at the panel, please do consider my application for this award. Wassalamu alaikum wa rahmatullahi wa barakatuh.